Thank you very much, Dean Eddington, for that extremely kind introduction. I am very happy to be a partner, and CEDAR is really happy to be a partner with the school of pharmacy here. It's been a really productive relationship. So the question is, why are we gathered here today, and what is the framework? I'm going to step back a little bit and talk about the framework for how did we get here and what are we trying to accomplish. And that gets to the point of why is FDA involved in biomarker development? When I took over CEDAR in 1994 for the first time, actually there had been a turmoil around drug development and biomarkers at that time because that was in the middle of the HIV crisis. And biomarkers had again surfaced as far as we need a surrogate endpoint, we need a biomarker, what are we going to do? And steps had been taken, and I'm going to talk about that a little bit. But my point is there is a intersection of the actual drug development with the science of understanding human responses, which relies to a great extent on biomarkers, and accept the empirical clinical methodologic science. So we've long accepted at FDA the use of biomarkers. Obviously, they're used ubiquitously in clinical medicine and in clinical trials, right? So they've long been used in drug development, and they've been used for everything from diagnosis, enrichment, monitoring, and of course the surrogate endpoints. But the FDA over the years, and speaking back in the last century, mostly depended on the community to work these up, okay? And eventually we'd get, in my mind, we'd get used to them enough, everybody in the clinical community would be used enough to the biomarker that they'd say, well, okay, community's accepted it, we have this amount of studies, and it's been used a long time, so we're going to accept it too. So there wasn't really a process. It was like the FDA waited quite a while. And in many cases, if you think of prostate-specific antigen, we waited forever. Okay, it's been a really long time. For some uses, it has never actually, no one's ever really figured out how rigorously, and that is reflected in the controversy about it right now. All right, nobody's really figured rigorously out what it means in various contexts of use, and so FDA really, say in cancer world, never really accepted PSA in the way many oncologists urged us to do. So when used as a surrogate endpoint, biomarkers were accepted much more slowly. Obviously, there's a checkered history around the use of various biomarkers as surrogate endpoints. Blood pressure, which was adopted based on sort of plausibility plus epidemiologic data, worked out really well. And the community was kind of on a roll about that, and then they got into the cardiac arrhythmias and the experience of the CAS trial and so forth, and that really soured the community and led to a lot of suspicion and skepticism about the use of other than outcome measures. And the cardiovascular community to this day uses outcome trials primarily because of their bad experiences with biomarkers. But then HIV, as I said, and subsequent adoption of first CD4 and then viral load was really successful, even though it didn't meet neither of those biomarkers met all the strict criteria that had been thought through about how you might qualify and accept a surrogate for efficacy. But it was successful and led to successful treatment. But then more recently, of course, we've had the HDL saga, and that everyone knows about. And so again, we have continued guarded skepticism about use of surrogate markers, surrogate endpoints as biomarkers as surrogate endpoints. So, but overall, if you look at the entire field, the development of biomarkers into drug development has been quite sluggish. And this is because, I think, regulatory skepticism about this. companies would come to the FDA with proposals frequently, and still do, to use this biomarker, rely on this biomarker, that biomarker, not necessarily for a surrogate endpoint, but simply reliance in some part of the development program. But often there isn't enough data, because one company, it's very difficult to generate enough data about a biomarker. There isn't the public vetting, the scholarly vetting, the general acceptance, and FDA has often said, no, we're not going to be able to utilize that particular biomarker. And to exacerbate this situation, really, and one of the crux of the issues in my mind, is that academics could and can to this day get funding to discover new biomarkers and publish those findings, but they can't get funding usually to do the work to develop robust data about the performance of the biomarker and understand the performance. You know, I think it would be really hard to put in a grant to perform analytical validation and get a good score on that grant, or even the clinical correlation studies, which can be very expensive. And so this has put a block in the science, because it's hard to move those thousands of biomarkers that are discovered further down the path into clinical reliance on the biomarkers, because there isn't a funding pathway to evaluate those. And this is a non-trivial exercise, as the people who are here from CPATH will know. It isn't just a matter of assembling a few data sets and waving your magic wand over them. There are many barriers to getting this done. So it seemed that really, to advance the field of biomarkers, one company or one sector, say the academic sector, there wasn't really a pathway to advance biomarkers through any given sector. And none really had the resources that were provided to them to do these activities. And so we worked under the Critical Path initiative that Dean Eddington mentioned, to put together consortia to do this. And we figured, okay, it would take a village, okay, so if everybody would contribute a little, then perhaps we could get some of these biomarkers developed and develop the evidence we needed. But the first thing we found, we started doing this probably sometime after 2005, we started working on this. And the very first thing we found was that even for the low-hanging fruit, because naturally that's what we tried to identify, what biomarkers are just almost there and over the finish line, there were competing assays in different academic centers. And there was little analytical validation of those competing assays. And the acquisition protocols for the samples varied. And so then we were told, well, just put all this data together from these different centers, and then you'll have the story. And we didn't have the story. We had 4 or 5 different stories. And in one case, early with the Biomarker Consortium, I believe David Woolley's here somewhere, we had the Lymphoma and Leukemia Society provided us funding, and basically we locked a bunch of PET scanners and people in a room for a day and forced them to come up with a single image acquisition protocol. It took a whole day, quite a bit of coffee. But this, it's a sine qua non, it's a baseline. You can't just keep doing different protocols all the time and acquire different images and then put it together and get something out of it. So we learned that very early, and I think the CPATH and the Biomarker Consortium learned that, CPATH Institute learned that. We're going to have to start with the basics here. We're going to have to start with standardization of practices, sample preparation, some of the things you're going to talk about today, and then analytical validation of a method. It really shouldn't be used broadly in clinical studies and everything if it hasn't been well validated analytically. So so this led to more work that had to be done. So the task, even for the low-hanging fruit, was more serious than we thought and would require more effort. Another thing we found that there was no common vocabulary among the different disciplines working in the field. And so I think the clinicians would talk past the diagnostic people, and people were not able to, what is a prognostic biomarker, response biomarker, this kind of biomarker, that biomarker. And the lack of this common definition and common language impeded common understanding of what we were trying to accomplish. And for these consortium, where the objective was regulatory acceptance, it was also difficult to say what the goalpost was. So what do you have to do to achieve regulatory acceptance? Because obviously it's going to be different from a prognostic biomarker and this and that. And that's where FDA, when we started the biomarker qualification process, we started talking to people in that process. We came up with the idea of context of use, right? So instead of saying it's a prognostic biomarker, what do we say? What exactly are you want to use it for? What decision do you want to make with this biomarker? What decision will this contribute to? And that then drove the goalposts, what kind of goalposts you had to get over. And that was a very specific way of talking about it. However, the words context of use are not immediately accessible to every community. And I bet a lot of these people in this room don't really know what I'm talking about right now, except I just explained it, right? So we definitely need to work on the definitions, whatever you want to call them, the taxonomy, the mutual understanding of these things better. Because I think what our qualification staff has found is people come in, try to get these qualified. We have to have an extended back and forth conversation about what are you going to use it for? And often people haven't really thought that very well. They want to qualify this analyte, this biomarker, and they haven't really thought about, okay, what are the situations, what decisions will this support? And this is very important. But then the goalpost was, okay, if you have figured that out, the goalpost is, what do the regulators require for supporting that kind of decision? What kind of evidence do the regulators want? If you've decided what kind of decision, okay, if you're going to decide your decision is, this is safety of the brain vasculature, okay, and this is a biomarker that even a drug that we're very worried about causing cerebral vasculitis, we can monitor this test, and then we'll keep giving the patient the drug because we think, okay, this test is very sensitive for the development of cerebral vasculitis, right? So how much evidence do you want to be confident that you can do that? And the secret is we didn't know either, okay, because we weren't in this business. The FDA wasn't in the business of approving biomarkers for drug development. We were in the business of approving drugs for marketing. And so my point is we're all learning together. We have learned a great deal, I think, in the last 8 years in this conversation we've had. However, it's time, I think, to really get down and get an evidentiary standard established, get a taxonomy established, get a common vocabulary so we can all talk to each other about this without having these long preliminary archaeological expeditions about what are you trying to prove here, what you want to support, what kind of decisions are you talking about. And but the fact that we didn't know either at the time, I think, led to some fumbling around both on the outside and on the inside as we tried to figure out, to give sensible advice to people about what kind of evidence would be needed. And frankly, for the FDA reviewers, I think this was quite difficult. They are used to making decisions, marketing decisions, are extremely momentous decisions. Is a drug, are millions of people in the United States going to be able to be exposed to this drug? Okay. That's probably not the same kind of standard you'd apply to an enrichment biomarker, right? But what other standard is there? There was no other standard articulated, right? So, and we didn't help our reviewers that much either, I don't think, because it's just go try this, nobody really knows, try it, and we'll see what we come up with. So, the hope we had for consortia, and that hope still remains, is that biomarker studies could be piggybacked onto clinical trials that are being conducted for other purposes, right? because clinical studies are very important and expensive, but there are many going on. And often to do the biomarker correlation work, you're going to have to do work in the clinic. You're going to have to do clinical studies. And this, so the hope was, once the analytical validation and the other necessary steps had been accomplished, and you had an idea of what kind of documentation you needed, what those goalposts looked like, then you could identify clinical trials that consortium members were conducting and then piggyback biomarker evaluations onto those. That has the benefit often, if they are intervention trials, you can see how the biomarker moves also with an intervention, not just it is better in some ways than just a natural history study with the biomarker. However, we still have the issue of the evidentiary criteria, the goalposts that remain, and I think is the focus of this workshop. So both the CPATH Institute and Biomarker Consortium, and actually other consortia that are ongoing, have managed to figure out how to piggyback onto clinical trials and how to gather that data and so forth. But no one has really clearly articulated the evidentiary criteria that would match up to a certain type of biomarker. But there's a certain overarching framework that I think we're all agreed on and that I'd like to talk about a little bit in the few minutes remaining. The certainty that we must have in a biomarker's predictive or diagnostic value is related to what it's going to be used for and thus the risks involved in depending on it. So like the example I used for cerebral vasculitis, it depends on the stakes. What are the stakes involved in depending on that the information from that biomarker, making decisions about human health in clinical trials as in drug development based on that biomarker? And that's where understanding the context of use is so important and getting some names or labels for those contexts of use. For example, if a biomarker is used in an enrichment strategy, okay, so you want to put more people like this into the trial, okay, and maybe it's for prognostic enrichment or maybe it's for other kind of enrichment, okay, but the biomarker doesn't really perform well. So what does that do? That introduces noise, right? And that is, the trial, for example, may fail to show the effective and effective drug. That would be one of the risks from the noise, because you expected the biomarker to get people at very high risk or with this kind of problem, and you didn't because the biomarker didn't work very well. Well, then maybe your trial fails when it might not have. Now, we shouldn't downplay that. That's use of human subjects in a way that is futile, right? And that's a bad outcome, although it happens all the time. It doesn't cause tremendous harm to people, but there are stakes involved in using an enrichment strategy that is actually not enriching. So, but that risk really pales beside the risk inherent in trusting subjects' renal health or CNS function to a new safety biomarker. I would argue that safety surrogates, particularly if they're standalone, they're the earliest sentinel against that. You're trusting them to be the earliest warning signal against that harm, okay? They carry the highest evidentiary standard, I think higher even than a surrogate for efficacy, okay? So now the CPATH Institute has taken on some of developing new safety biomarkers in many areas where there's a need, okay, for that sentinel marker because we don't have other good testing strategies for early detection of drug-induced toxicity. But that's a high burden. That's a very difficult burden because we're going to be trusting that. Now, there are intermediate steps you can take in those, and some of those have been taken, but that's a kind of conversations I think that have to be had. Now, a lot has been written about the workup of surrogate endpoints that people call validation of surrogate endpoints. They call them different things. There are a lot of literature on this. surrogate endpoints for efficacy. But little has been said about the use of surrogate endpoints for accelerated approval. And that's where many of our new surrogate endpoints come in. The legal standard for surrogate approval, for accelerated approval, is different than using it for a regular approval. And that standard is that the surrogate endpoint has to be reasonably likely to predict clinical benefit. So we should all think about this. What does reasonably likely mean? And the FDA really needs to pick this up, and we plan to, and work it up in concert with the community. We have a lot of experience with doing accelerated approvals. And what is this lower standard than the full approval using a validated surrogate? What is it? You know, how much evidence do you have to have? We have not articulated that. I'm not sure we actually know. Now, there are many other types of biomarkers, as we all know. And we need a classification system that everyone buys into. I think FDA is working on that with NIH so that we can at least talk to one another, but we need the whole community to buy in, because if we talk about evidentiary criteria, as I said, they're inextricably linked to the use, whatever the use of that biomarker is. And you can't really separate those two things. So we need to be able to have a conversation where everybody understands what everybody's talking about. Now, because of all this, all these problems, of course, FDA a number of years ago set up a biomarker qualification process, and you're going to be hearing about that today. And we've been running that process, and I think we've qualified like 14 biomarkers, maybe. 14 biomarkers have been qualified for very specific contexts of use over the years that the program has been operating, and many others are in back and forth with the sponsors, which are often consortia. But this process has generated controversy because of all the problems I just went over. Okay. It isn't like people can come in like an NDA and they know what the standards are, and you come in with an NDA and we review it in a certain amount of time, and then your drug gets on the market if you have successfully shown all these different things that people know they have to show. It's quite different with biomarkers. I don't think we, and I don't think the community, fully understand what needs to be shown. And so when people propose a biomarker to us in the qualification process, there's a long back and forth. It might take years to talk about what are you going to use it for? What are the consequences of that, and so forth. And because of that, people are now very interested in biomarkers. That has gotten congressional interest. And so in the 21st Century Cures bill that was before the House, and the House voted out, there's mention in there of biomarkers, but we had a long discussion with the Hill staff about biomarker qualification in the process. They were very interested in that. And I expect the same thing will play out in the Senate as well. So, but the academic community is relatively silent on this, I think, for the reasons I mentioned very early. There are no grants to be had for biomarker qualification. And of course, academics can only work on things that have a possibility of getting funded. So with various institutes that are going on, we're trying to gather different sources of funding to provide that support for this process. However, it's quite possible Congress will intervene in various ways, and I expect this will be a topic of discussion in the next prescription drug user fee negotiation that goes on as well, because the industry, I think, is realizing that qualification of biomarkers could help move the field along. In fact, there's recently been a great deal of interest in precision medicine. What's precision medicine? Well, I've always said that Americans love to intervene, but in medicine, really diagnosis is the foundation, not intervention. And you're really supposed to know what you're dealing with before you start treating. Okay? So really, we need a whole generation of new, reliable, and informative biomarkers before we can do precision medicine. And so this has become more urgent, I think, as the era of molecular medicine becomes a reality. And back to this workshop, evidentiary criteria are really the heart of the matter. Once we get over what we're going to call them, the taxonomy, the classification, or whatever, then really the question is to rely upon this bit of information provided by this assay. How much evidence do you need that it's reliable? And how do you generate that evidence? Which is, in the next couple of days, there's going to be discussions of that in the next few sessions. So I think it's important to realize that FDA doesn't inherently know. Okay, this is something that has to be worked out by the community. We're participating together. We aren't repositories of knowledge in this field. We have worked on it for the last 8 years or so, and so we have quite an inkling of what might be involved. But there are many others. Even though this isn't highly funded in academics, we've had many academic researchers all over the country who have persevered on their assay over decades of their career, probably incorporating it like tangentially into their grant proposals so that they can continue to get it funded. So there is a considerable repository of knowledge out there. However, we all need to come together and really make this more of a scientific field of endeavor if we're going to actually deliver precision medicines. We've got to have precision diagnostics that work for those medicines that tell us what to do. So I expect you'll make progress today. I think this is a really good group. I thank University of Maryland and the CPATH Institute for co-sponsoring this. And as you think today, there are two things I think you ought to think about. One is what research program should be advocated for and should be taken forward, okay? Because ongoing scholarly investigation into these areas is what has been missing and what is needed. The FDA, we're not in a position to do that. We're not academia. And second, what can be done now? Can we develop provisional criteria? Can we do other things? But also, can we put down a research agenda and see how that can be accomplished? So good luck on your work today, and thank you very much for the opportunity to frame this issue.